Welcome to Click for Biology. We're carrying out plant experiments, particularly on the topic of plant nutrition. And in this particular video, we're going to demonstrate that carbon dioxide is required for photosynthesis. If you would like a copy of this presentation, that's as a PDF down in the description. You can click on that, download that, and annotate it with your own notes. So demonstrating that the plant requires carbon dioxide is carried out using these glass jars here, sometimes referred to as bell jars. In photosynthesis, carbon dioxide and water are combined in the presence of light and chlorophyll to make glucose and oxygen. And of course, we'll recall from other videos that glucose in a plant is quite difficult to detect because the plant joins the glucose molecules together into long chains and stores these as the molecule starch. Now, starch is quite easy to detect and show that it's present. So we're going to find starch as our indicator that photosynthesis has occurred. So we will begin, of course, first by destarching the plant for 36 hours in the dark. And so both of these plants will not have starch to begin with. The bell jars in the image are different. In this bell jar, the air inside the bell jar contains carbon dioxide. The bell jar is sealed with Vaseline at the base. In the right-hand jar, we have the same kind of plant. Everything is the same, except we have present what is called soda lime. And soda lime will take any carbon dioxide out of the atmosphere of the bell jar so that there is no carbon dioxide in the air of this jar. We would allow both plants to be exposed to light for perhaps a 24 to 36 hour period. And then we would carry out the test for starch, which we have demonstrated in the previous video. We would expect and predict that because this bell jar contains carbon dioxide, the carbon dioxide will have joined to the water in the presence of light and chlorophyll to make glucose, and the glucose will join together to make starch. So that when we carry out the starch test, we find that the brown color of the iodine solution has turned to blue-black. This demonstrates that you need carbon dioxide substrate for photosynthesis. The control on the other side will have removed the carbon dioxide, and so even in 24 to 36 hours, with no carbon dioxide, no glucose is made, no starch is made. And so when we test this leaf for starch, we find that the brown iodine solution stays brown because there is no starch present. Okay, let's do a 3-minute quiz on the content that you've just listened to. Pause the video, read the questions, pause the video, write down your answers, and then go down to the description in the video, and you'll find the answers there so that you can check your progress. Having done that, you can move to the next experimental technique, which is linked as the next video here. We have the entire playlist linked, the channel so that you can subscribe, and a thumbs up for this particular video, please. Thank you for listening.